Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today we are going to talk about another molecular markers that is called as restriction fragment length polymorphism. In abbreviation we are going to call RFLP. So first we will say what is genetic markers or molecular markers or DNA markers. Genetic markers is a gene or DNA sequence which normally present in on our chromosomes at a specific locations, a specific place, and that help us in the identification of any individual or any species. So we identify, we separate, we discover two different organisms, two different species on the basis of the presence of that specific location on the chromosomes. So it is described as a variations. So molecular markers normally provides variations. So different organism will have different kind of gene sequences on their chromosomes. Now there are different type of molecular markers we are having: RAPD, RFLP, AFLP, SCAR, SSR, SLP, SNP. Number of things are there. Already we have talked about RAPD. So that is random amplified polymorphic DNA. Today we are going to talk about RFLP. So what is RFLP? RFLP is abbreviation of restriction fragment length polymorphism. In pronunciation we are going to call it RFLP. So if you see the name, it is called as restriction fragment length polymorphism. Means in this case we are going to use restriction. So first what is this here? This is a techniques which normally exploits the variation in the homologous DNA sequence, that is called as polymorphism. So the different kind of variation, different kind of differentiation in the homologous gene sequences, that is called as polymorphism, and that normally different in different kind of individuals, different populations, different species. So on the basis of this polymorphic DNA which is present on the chromosome at a specific location, we are going to differentiate it. So what is this polymorphism? So it is a kind of variation in the DNA sequence which is normally recognized by restriction enzymes. Now what is restriction enzymes? Restriction enzymes are, it is a kind of endonuclease which normally recognize DNA at a specific sequence and then cut the double-stranded DNA at both the strands. So that is called as restriction enzymes. Normally these restriction enzymes scientists have discovered from the bacterial cells. So they isolate, like EcoR1 we are having, BAMH1 we are having, HIND3 we are having. So different kind of restriction endonuclease we are having. So here we are going to use this restriction sites which is present on the DNA sequence for the differentiations. So RFLP is used as a marker on the genetic maps, and then it will be separated on the gel electrophoresis, and then we can visualize it on the gel. So this is the sequential events how RFLP can be performed. So it starts with DNA extraction. So first DNA will be extracted from the organism, from the individual, from the species what you want to detect it, and then that DNA is going to be spliced with the restriction endonuclease. So it will be digested by restriction endonuclease at a specific sequence. So it will be, you will have a different DNA fragments. That different DNA fragments is going to be isolated, going to be separated on the electrophoretic plates or electrophoretic gels, depending on the sequence size and other thing. So it will be separated. So we will have different kind of bands. Now these different bands we are going to transfer onto the nitrocellulose membranes through southern blotting techniques. So it will be transferred onto the nitrocellulose membranes. Once it will be transferred, it will be blotted onto the membranes. Then it is going to be hybridized with the labeled probe. So it will be visualized, and then it will be through autoradiography you can see it, like you can see in the picture. So first this is a DNA sequence which we want to identify. So we have, this is from an abnormal case, this is from the normal person. So if you see in this case, in the case of normal sequence, there are different restriction sequences are there. So when, and in the case of disease, because this site is mutated, so the third sequence is not present here. So the second, this DNA is going to be digested from here, it will be digested from here, it will be digested from here. So you will get total 1 fragment, 2 fragment, 3 fragment and 4 fragment. But when this second DNA will be, this diseasic abnormal mutated DNA will be going to be spliced with this restriction enzymes, it will give you 1 fragment, this one, because it will be digested from here, and second one piece will be this one, and third fragment will be this one. So after this splicing, you are going to do the gel electrophoresis. So you will have different bands from the normal, also from the abnormal, which you want to match it, and then it will be transferred through the southern blotting on the means nitrocellulose membranes. Through then it will be associated means hybridized with the probes and visualized through autoradiography. So ultimately you will have the specific place you can visualize it. So you can see in the whatever is, means in this case, whatever DNA is present in the normal, same DNA sequence, same DNA fragment is present in the abnormal DGG case also, except few bands. Like this band is absent in the case of DGG abnormal case. This DNA is absent in abnormal case. This DNA is abnormal in the normal case. So this is a mutated DNA. So that 3 bands you can see, which is difference in all the 3 cases. Now there are number of merits and demerits are there. One thing you have seen it, compared to RAPD, here we are not going to use PCR. So this is not depending on the PCR. Here we are going to use DNA probes. Here we are going to use southern blotting. Here we are going to use different kind of autoradiography, but not the PCR. So what are the applications where we are going to use this RFLP techniques? This is normally used for the status of the genetic disease identification, like some of the disease which is caused because of the mutations, gene mutations. So that DNA can be, mutated DNA can be detected by this RFLP techniques. It also helps us in the determination of or confirmation of a source of DNA sample, such as in case of paternity testing or criminal investigation. So these 2 examples we will see in the further slides, how we can go for paternity testing, how we can do the criminal investigations. Then its third important use is genetic mapping to determine the recombination rates, which normally show the genetic distance between the other loci. Fourth, and another example is you can identify a person who is going to carry a disease causing mutation in their genes. So any kind of mutated genes which is present in a family, that can be detected. So these are the different applications, but the abnormality of this technique is RFLP normally going to give you, it requires many steps, as you have seen. First, splicing, DNA extraction will be there, then restriction splicing will be there, then you have to separate with the gel, then you have to transfer onto the nitrocellulose membranes, then you have to do the hybridization with the probes, and then autoradiography. So multiple steps are there. If steps will be more, so chances of doing mistakes will be more. So abnormality or any kind of results will be affected in that means ratio also. So more steps, more abnormality, more mistakes. While other techniques, which similar type of things can be done with the PCR based techniques also. So there you don't have many steps and less time also, because in this case we require many means time, like blotting and other things. It can take days, but in the case of PCR you can do same test by within a hours. So that is disadvantage of this RFLP techniques. Another disadvantage is, in this case, this technique is not based on PCR. So we require a large amount of DNA, and for the separation, for the procurement of large amount of DNA, we require, it is a time consuming process. But PCR based techniques can give you within minutes a large amount of DNA. So that is little bit easy techniques, which is comparatively similar, going to give you similar type of results. Then there are due to numerous reasons, whatever we have already discussed, PCR techniques are normally going to replace RFLP. So RFLP nowadays normally we are not going to use many places. So in place of RFLP we are going to use PCR techniques, like PCR, paternity testing and forensic science and analysis, and number of other things. Another drawback is the identification of SNP in case of human genome project has almost replaced RFLP for normally when we are going to detect any disease because of mutations. So mutated genes, any kind of such disease status analysis, normally we are going to perform by SNP analysis rather than going for RFLP technique. So these are the different disadvantages. Now we will see 2 important examples how this RFLP is going to be used for criminal investigations, DNA fingerprinting techniques, and paternity detections. So first, in case of DNA fingerprinting, if you see, this is a victim, and from the crime site, crime scene, this DNA sequence, these are some DNA sequence has been got. So those DNA sequence has been fragmented and jail has been run, and these are the three suspects. Now I have to find who is the actual criminal, whose sample we have found at the crime scene. So what we are going to do, we are going to see the match the gene which is means of DNA fragment from the crime scenes with the victim and with the suspects. So we can see which of the victim, which of the suspects is going to match the maximum bands. A maximum similarity is there. It might not be necessary that 100% similarity, similar result will be there, but the maximum DNA fragment will be of the crime scene will be matching with the suspects. So if you will see here, in case of suspect 1, this DNA is not going to match here. This DNA is not present at the crime site. Similarly, this DNA segment is not present in the sample which is taken from the crime site. In case of suspect 3, this DNA fragment and this DNA fragment is absent in the crime scene. But if you see suspect 2, all the DNA fragment, this one, this one, this one, this, all the DNA fragment which is sample taken from the crime scene and the suspect 2 is maximum matching. So it means the suspect 2 is the criminal. So this by this RFLP techniques or by using restrictions and jail references, you can detect the criminals. Similarly, when we are going to do paternity testings, like this is the DNA fragmentation done from the child's baby DNA. So, and this is the DNA fragment from the mother DNA, and these are the two suspected father. So again, we are going to see match the child's DNA, whatever DNA fragment will be present in the child, it should come either from the mother or from the father. So like you can see here, this DNA of the child is coming from father number 2, suspect father number 2. This DNA is also coming from father number 2. So and this is coming from mother, this is coming from father number 2, this is coming from mother, this is coming from father number 2. This 2 is not matching anywhere. This is matching with father number 1, this is from mother, this is father number 2, this is mother. So if we'll say maximum match is from the father number 2. So possibility is father, who is the suspect number 2, this is the possibly father of this child. So on the basis of these restriction and then gel electrophoresis, hybridization and probe detections, autoradiography, we can detect the criminals and we can going to for the paternity testing. But nowadays this RFLP testing, RFLP marker is not going to be used very much, rather than PCR based techniques is going to be used mostly. So this is all about the RFLP. Next day we will come with the another genetic marker, that is AFLP. So thank you very much. Have a nice day. If you are not subscribed my channel, please subscribe it.